Hello everybody, it's your friend Daniel here and welcome back to another past paper video. In this video we're going to be covering the specimen paper for AQA GCSE Biology paper 2. And without any further ado, let's jump straight in. Now, question 1. Charles Darwin proposed a theory of natural selection. Many people at the time did not accept this theory. There was a different theory at the same time as Darwin's theory. This different theory said the changes in an organism during its life could be inherited. Who proposed this theory? Now this is just something you need to remember from your textbook, not really any logic behind this question, just something you need to memorize. And it is Jean-Baptiste Lamarck. Moving on to question 1.2. Studying fossils helps scientists understand how living things have evolved. Figure 1 shows a fossilized snake. Explain how the fossil in figure 1 may have formed. The snake is covered in mud. Then the soft parts decay while the hard parts, which are the bones, don't decay. These bones are then replaced with minerals, forming a fossil. Now moving on with question 1. There are many types of rat snake in the world. Table 2 shows two types of rat snakes. The type of snakes are the Japanese and the Texas rat snake. The Japanese rat snake is green, while the Texas one is pale brown. The Japanese one has a grassy environment, while the pale brown one has a dry and dusty environment. And the question related to this is to explain how the Japanese rat snake evolved to be different from the Texas rat snake. This is my answer. It's a bit long, but I'm just going to go through it and then explain it a bit. And this is due to there being extensive colour variation in snakes caused by different genes, alleles, or mutations, and among these exist multiple shades of green, some of which more closely match the local environment of Japan than others, providing superior camouflage. And the snakes being so well concealed allows these grass snakes to catch more food and avoid being eaten more effectively than individuals whose colour stands out. This allows the best camouflaged survivors in each generation to breed and produce offspring that will inherit the genes and mutations responsible for the advantageous shade of green, gradually shifting the population towards even better environmental matching and a particular shade of green. Now this may seem like a complicated answer, but you're just applying the theories of natural selection to a particular colour of snake. And as time goes on, the snakes which most match the environment of Japan will survive more until the snake almost perfectly matches the shade of grass that is found in Japan, allowing them to be good predators. And 1.4, many species of snakes have become extinct. Give one reason why a species might become extinct, and this is due to a new predator. Now moving on to question 2. A gardener wants to add compost to the soil to increase his yield of strawberries. The gardener wants to make his own compost. And the question for this, starting off with 2.1, is an airtight compost heap causes anaerobic decay. Explain why the gardener may be against producing compost using this method. And this is due to methane being produced from the anaerobic decay, which is a greenhouse gas, which could cause global warming. Moving on. The gardener finds this research on the internet. A carbon to nitrogen ratio of 25:1 will produce fertile compost. Look at table 2, and we're given a list of type materials into compost, and then the mass of carbon and nitrogen in each sample and the ratio. First things first is to determine the ratio X in table 2. Now you could do this by just doing 9.8 divided by 0.2 equals 49, and that being a ratio of 49:1. Or you could set up a ratio of 9.8 to 0.2, and then just multiply both sides by 5 to get 49:1. And just do whatever method you're most comfortable with, and whichever method you usually use for your GCSE mathematics. And then for 2.3, which type of material in table 2 would be the best for the gardener to use to make his compost? And this is because horse manure, because as you can see, horse manure has a carbon to nitrogen ratio of 20:1, which is the closest to the, as stated on the internet, perfect 25:1. And moving on to 2.4, this is a 6 marker. This is all the indicative content points for the 6 marker. It's a bit ridiculous. Some of the leaves from the gardener's strawberry plant die. The dead leaves fall off the strawberry plant onto the ground. The carbon in the dead leaves is recycled through the carbon cycle. Explain how the carbon is recycled into the growth of new leaves. Dead leaves are broken down by microorganisms respiring, and therefore releasing the carbon from the leaf structure as carbon dioxide into the atmosphere. When plants photosynthesize, they use carbon dioxide to produce glucose, which is then used to make proteins for the growth of new leaves. Now, it's a bit ridiculous for 6 marker that there's this few content for it. Of course, this is a specimen paper, meaning no one actually sat this, but it is a fairly simple 6 marker to just explain the carbon cycle. Like also my actual things, like if we size it up to take 1 line each, it actually takes a lot more space. So that is what it would look like in an actual thing. Still not that much space though. You would expect more. Okay, now moving on to 2.5, figure 2 shows 2 strawberries. Both strawberries were picked from the same strawberry plant. Both strawberries were picked 3 days ago. The strawberries were stored in different conditions. Give 3 possible reasons that might have caused strawberry A to decay. Now, the 3 possible reasons are strawberry A was stored at a higher temperature, strawberry 2 was stored in a higher oxygen environment, and strawberry 3 has more moisture. Now, moving on to question 3. Many different types of animals are produced using selective breeding. Some cats are selectively bred so they do not cause allergies in people. Suggest 2 other reasons why people might selectively breed cats. And one of these reasons is for aesthetic reasons, such as fur colour. And another one is to produce docile cats, which are not aggressive. And selective breeding could cause problems of inbreeding in cats. Describe one problem inbreeding causes, and that is that cats are more likely to be susceptible to diseases. Next up is a bit of a longer question with a bit of preamble. Many people have breathing problems because they are allergic to cats, called FeLD1. Different cats produce different amounts of FeLD1. A cat has been bred so it does not produce FeLD1. This cat has been produced using selective breeding. I just realised it may be FeLD1, like feline. Yeah, either way. Questions unaffected. Parents with the desired characteristics of low amounts of FeLD1 are selected. The parents are bred together to produce offspring. In that offspring, there will be individuals with different amounts of FeLD1 produced. The offspring of the lowest amounts of FeLD1 are selected and bred. This is repeated over many generations. In each generation, the lowest FeLD1 producing individuals are chosen over time, producing offspring with less and less FeLD1 until you reach a cat, that's like in the question, that does not produce FeLD1. Moving on to question 4. A student plans an investigation using mold. Mold spores are hazardous. Give one safety precaution the student should take when doing this investigation. Wear a face mask. A student made the following hypothesis about the growth of mold. The higher the temperature, the faster the growth of mold. The student planned to measure the amount of mold growing using the following equipment: slices of bread, plastic bags, knife, chopping board, mold spores. And then we're asked to describe how the materials and equipment could be used to test the hypothesis. This is a fairly standard design and experiment question. And firstly, you cut a specified number of pieces of bread to the same size. Then place mold spores on the bread. The number of mold spores need to be the same quantity of mold spores on each piece of bread, and then you continue and place bread in different sealable plastic bags in different temperatures, e.g. a fridge, which would be around 3 to 5 degrees Celsius, a room which has room temperature of around 20 degrees Celsius, and an incubator which have a temperature of around 40 degrees Celsius. And then you leave each piece of bread for the same amount of time, such as 4 days, and you measure the percentage of mold coverage on each bread. Now I've just put something that it's accountable in every experiment. It just does a bullet point. There's only one thing that really applies that's in every single experiment, and that is to repeat the experiment to reduce uncertainty. Now for 4.3, you're meant to give one variable that the student should control in this investigation, and that is the type of mold. For example, you wouldn't want to use different molds for different temperatures, as different molds may grow at different rates. Then we're asked to determine the rate of mold growth at 42 degrees Celsius between day 2 and day 7. Now, because it's 42 degrees Celsius, we're using the straight line. And firstly, we need to work out the change in the amount of mold units, because it goes from 4 to 56, that's 52. And then you divide 52 by the amount of days, which is 5. 52 divided by 5 is 10.4. So the rate of mold growth is equal to 10.4 units per day. Mold growth shows decomposition of the bread. Give a conclusion about decomposition from the results in figure 3, and that is that decomposition occurs at a faster rate when temperature is higher. Moving on to figure 5, we're shown reflex in the iris of the human eye in response to changes in light levels. Describe the changes in the pupil and iris going from A to B in figure 4, and we're asked to explain how these changes occur. And we're asked to refer to the changes in light level in your answer. And this is the pupils are dilated in image B, meaning there are low light levels because the circular muscles in the iris relax and the radial muscles contract. Once again, telltale sign this is a specimen paper, with there being such few informations that would get you all 4 marks. Then for 5.2, figure 6 shows light entering the eye in a person with blurred vision, and figure 6 shows how this condition is corrected with lenses. We're asked to compare the figures and explain how the blurred vision is corrected. Now, for figure 5, it shows refraction, where light does not focus on the retina, which would be around this point here. And in figure 6, the lens bends the light, so the light focuses on the retina, causing correct vision, while in figure 5, there would be blurred vision because the light does not correctly focus on the retina. Then for question 6, two students investigated reflex reaction times. This is the method used. Student A sits with her elbow resting on the edge of the table. Student B holds a ruler with the bottom of the ruler level with the thumb of student A. Student B drops the ruler, and student A catches the ruler and records the distance as shown in figure 7, with steps 1 to 4 then being repeated. And then we're just shown a ruler and shown how it's done. And we're asked to suggest 2 ways the students could improve the method to make sure the test would give valid results. And this is to drop the ruler from the same height each time, and let the ruler drop without using any force, e.g. like just drop it and let go instead of throwing it downwards. When my class did this experiment, when I was doing my GCSE biology, everyone just sort of threw it downwards because they wanted everyone else to have bad reaction time, because they wanted to have the best reaction time, and it just completely messed up the experiment. Don't do that. That's basically it. Then for table 3, we're shown students A's results of different test numbers and the distance the ruler dropped before it was caught. And we're asked to work out a median result. And what you need to do is you need to put all these numbers in order and then just find the middle number, which is 117. And then the mean distance the ruler was dropped was 160 millimeters. Nice of them to give that to you. And we're asked to calculate the mean reaction time. And we're asked to use the equation reaction time in seconds equals square root of mean drop distance in centimeters divided by 490. And we're asked to give our answer to 3 significant figures. Now what we do is we just plug it into the formula, convert 160 millimeters into centimeters, 11.6 over 490, square root equals 0.1539. However, because we're asked to give it to 3 significant figures, we give it a 0.154. The students then measure students' A reactions time using a computer program, and then they're shown a method. And using a computer program to measure reaction time is likely to be more valid, and give 2 reasons why. One indication is there's no indication beforehand when the colour will change. For example, just before the ruler is let go of, you could see that they're about to release it if you look carefully at their thumb. However, you won't be able to see on a computer when the colour is going to change, and that the measurement of time is more precise than reading from a ruler. Moving on to question 6.5, a woman has a head injury. Her symptoms include finding it difficult not to name familiar objects and not being able to remember recent events. Suggest which part of her brain has been damaged. Suggest which part of his brain has been damaged, with this being the cerebellum. Figure 8 shows an image of a small section of DNA, with Figure 9 showing the structure of that section. What is Part B, with Part B being a phosphate? 7.2. In Figure 8, the structure of the DNA shows 4 different bases. There are 4 different bases, and they always pair up in the same pairs. Which bases pair up together, and that is adenine and cytosine. Syndrome H is an inherited condition. People with Syndrome H do not produce the enzyme IDUA. Figure 9 shows part of the gene coding for the enzyme IDUA. Strand J is from a person without Syndrome H, and Strand K is from a person with Syndrome H. Explain how the mutation could cause the enzyme not to work. The mutation changes the C to a T, as you can see here, altering the DNA code with this change in the amino acid. The change in the amino acid causes the protein to change, so it forms a differently shaped active site. Therefore, the enzyme no longer fits the substrate, preventing it from working. It's a bit ridiculous that you can get all 5 marks with just this little written bit here, but it is a specimen paper, and AQA does not produce the best mark schemes. Now moving on to 7.4, a recessive allele causes Syndrome H. A heterozygous woman and a homozygous recessive men want to have a child. Draw a Punnett square diagram to determine the probability of the child having Syndrome H. Identify any children with Syndrome H. Use the following symbols. A equals the dominant allele. A capital A equals the dominant allele. A equals the recessive allele. And as you can see, for the woman, it's capital A and little a because they're heterozygous, which means it doesn't match. And for the man, it's both little a, which is the recessive allele. And because it's caused by a recessive allele, both need to be recessive for them to have the syndrome, which means double little a has the Syndrome H, and as you can see, I've identified them by circling them. And we're asked to calculate the probability being 0.5, because 2 out of 4 of the possible combinations have Syndrome H. Now moving on to question 8. Food security is when a population has enough food to stay healthy. Lack of food security is a global problem. One way to maintain food security is to increase the efficiency of food production. Figure 10 shows how some pigs are farmed using intensive methods. Now for 8.1. Some people think the farming methods shown in Figure 10 are unethical. Suggest 2 other possible disadvantages of intensive farming methods, and this is that the diseases spread more rapidly, and it leads to an overuse of antibiotics. With an overuse of antibiotics, as you may remember from other topics, leading to more bacteria building up antibiotic resistance, and overall it being more dangerous for both the animals and humans to when they get bacterial infections, and overall making it harder to survive. For 8.2, explain how the intensive farming of pigs increases the efficiency of food production. They're kept inside and enclosed, so less energy and heat is lost in controlling body temperature. Less energy is required for movement because they're enclosed and they can't really move around as much. This therefore leads to more energy being available for growth, which means they'll grow more and need to be fed less, meaning it's more efficient for food because it's cheaper. A newspaper reported that food security is a serious problem in remote communities in Canada. This is because the Aboriginal communities are eating fewer traditional foods. One traditional food eaten by the Aboriginal communities in Canada is seal. Looking at table 4, you can see how it's decreased from 362,000 to 67,000, and we're asked to calculate the percentage decrease in the number of seals caught from 2004 to 2010. Now, you may remember from GCSE mathematics that the deep formula for percentage decrease is, or any form of percentage change, is the change over the original times 100. And to work out the change, we just do 362 minus 67, or just 362,000 minus 67,000, it doesn't make a difference, equals 295, and then 295 over 362 times 100, with you getting a percentage decrease of 81.5. 8.4. The conclusion in the newspaper might not be correct. Suggest 2 reasons why. This is due to Aboriginal people being able to eat other foods, and just because their traditional food sources are at risk and are depleting, it doesn't necessarily mean that their overall food security is decreasing because they can still eat other food. And another point is that we do not know if other traditional food sources besides the seals have declined. Homeostasis controls the internal conditions of the body. Explain how blood glucose levels are controlled in the body of someone who does not have diabetes. And if the blood glucose levels are too high, insulin is released from the pancreas, so glucose is moved into cells and stored. And if the blood glucose levels are too low, glucagon, important not to get those confused, is released from the pancreas, which causes glycogen to be converted to glucose and released into the blood. What it's important to remember, as I for half a second did not do there, is that glucagon is the enzyme that converts glycogen into glucose, and glycogen is the stored version of glucose. Compare how each type of diabetes is caused and suggest how each type of diabetes can be treated. For type 1 diabetes, not enough insulin is produced, whereas the cells of a type 2 diabetic do not respond to insulin. Type 1 diabetes is treated with injections of insulin, whereas type 2 is treated with diet and exercise. Now the issue with this question is it says compare how each type of diabetes is caused, and then the half first half of the mark scheme focuses in the difference between like the two types of diabetes. It's not really focusing on causes of the diabetes, more so talking about how the different types of diabetes affect the body. Now for 9.3, looking at table 5, the population of the UK, we're given the population of the UK in 2015, the number of people diagnosed with diabetes, and the number of people to be estimated to have undiagnosed diabetes, and we're asked to calculate the percentage of the UK population estimated to have diabetes and give it two significant figures, which I have not. See, it's very easy to make mistakes, and it's okay to make mistakes as long as we try our best and get marks on the harder questions. Okay, so firstly, we're asked to include both diagnosed and undiagnosed people in our calculations, which is a bit statistically fraudulent, but this is biology, not statistics, so I'm not going to oppose to it. And so firstly, we add the amount of people diagnosed and undiagnosed together, which you can see I'm doing here, to get 3.999 times 10 to the power 6. And then we just use the normal percentage formula, 3.99 times 10 to the power 6 over the total population, which is 6.5 times 10 to the minus 7, times 100 to get 6.2%. Now, a urine test can also be used to check for the presence of glucose in the urine. Diabetes can also be diagnosed with a blood test to measure the concentration of blood glucose. Suggests why a blood test is more reliable than a urine test. And this is because there could be other reasons for glucose in urine besides diabetes. I don't like this question personally, because it doesn't say presence of glucose is linked to diabetes. Now, you can obviously work it out from context, but you shouldn't be expected to. They should either be asking you about that type of information, or they should be giving you that type of information. You shouldn't just be able to have to work out what the question means. I'm just disappointed in AQA about the quality of their papers overall, and especially the specimen papers. They're very low quality. Now for 9.5, a blood test called a glucose tolerance test checks how well the body processes glucose. Concentrations of glucose in the blood are measured before and after drinking a glucose drink. Patients are not allowed to eat food for 8 hours before the glucose tolerance test. Suggests why patients are not allowed to eat for 8 hours before the test. And this is so the results are not altered and not affected by glucose from the food that they could eat for 8 hours before. And then we're showing a little graph of the test, showing it for two patients, A and B, and the blood glucose concentration in units and glucose drink taken. We're asked which patient has diabetes, and it's patient A, and this is because their glucose levels remain much higher than patient B and remain high and do not fall over time, which is a sign of diabetes because either the cells aren't able to properly use insulin to absorb the glucose, or their body isn't producing insulin to turn the glucose into glycogen normally. Moving on to question 10. Endocrine glands produce hormones. Hyperthyroidism is caused by an overactive thyroid gland. Suggest what would happen in the body of a person with hyperthyroidism. And this is the fact that too much thyroxine is released into the blood, which raises the BMR, also known as the basal metabolic rate, which causes an increase in the formation of glycogen, lipids, and proteins.
Hi, I'm Laurie, and I'm going to be running through the last few questions of the paper. Question 10.2 is a pretty simple one. It asks you to describe the roles of FSH and LH in the menstrual cycle. The best way to answer this would be to say their roles are that FSH causes the egg to mature and stimulate ovaries to produce estrogen, while the LH stimulates the egg to be released. Moving on to question 10.3, the last question of the paper. This question provides lots of information, so first let's break it down to the necessary information. There are two main contraceptive pills, the combined pill, which contains progesterone and estrogen, and the mini pill, which contains only progesterone and has to be taken at the same time each day to be effective in preventing pregnancy. The mini pill is also usually worse at preventing pregnancy than the combined pill. So, this question wants us to explain why missing a dose of the mini pill would reduce the success rate of it, or in other words, be even less effective at preventing pregnancy than usual. Don't get bogged down in the wordiness of this question. First, break it down like we did just now, and then you're ready to answer the question. An ideal answer would go like this. Missing a mini pill dose causes a drop in progesterone. This lack of progesterone therefore leads to FSH not being inhibited anymore, with the lack of FSH then further leading to LH not being inhibited also. Therefore, an egg is matured and released, leading to an unwanted pregnancy. Well, that's the end of this paper. Make sure to like and subscribe. I won't see you next time, but Daniel will. I hope you do well in your GCSEs.